Good afternoon, one and all. A very warm welcome to all of you. I am Dr. Ramanlal Narayan Kachwe, Professor, Department of Pharmaceutical Chemistry, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, Nasik. As you already know, we have organized 1 week international e-FDP and e-STP on empowering pharmacy education, navigating regulations, global trends, and advancing outcome-based research. For this, we have wonderful personality, Dr. Rajendra Bhadane, Senior Scientist, Institute of Biomedicine, University of Turku, Finland. I take this opportunity to welcome today's guest, Dr. Bhadane, an expert in biocatalysis with ADP, ribosyl transferase and introduction to single particle protein crystallization for this online e-FDP. It's my great privilege to introduce to our second day, third session speaker of the 1 week international e-FDP and e-STP, Dr. Rajendra Bhadane sir. Sir has an 18 plus experience in research and academic. His educational qualifications, sir has completed Doctor of Philosophy, Gyan Vihar School of Pharmacy, Suresh Gyan Vihar, University, Jaipur. Master of Pharmacy in Pharmaceutical Chemistry, 2007, School of Pharmaceutical Science, Rajiv Gandhi Pradhyogik Vishwavidyalaya, Bhopal. Bachelor of Pharmacy in 2005, ERA College of Pharmacy, North Maharashtra University, Jalgaon. Sir is currently work in Senior Researcher, Turku Cellular and Microbiology Laboratory, Institute of Biomedicine, University of Turku, Finland. Sir has previously research experience in 2019 to 2022, postdoctoral researcher, Structural Bioinformatics Laboratory and Pharmaceutical Science Laboratory, Abu Academy University, Biocity, Turku, Finland. In 2016 to 2019, Acting Principal and Assistant Professor, Department of Pharmaceutical Chemistry, Divine College of Pharmacy, Satana, Pune University. 2010 to 2016, Assistant Professor in Department of Pharmaceutical Chemistry, Loknete J.D. Pawar College of Pharmacy, Manur. 2007 to 2010, Lecturer Pharmaceutical Chemistry, Department of Pharmaceutical Chemistry, NJMSPM College of Pharmacy, Anjaneri, India. 2010 to 2016, Assistant Professor. Sir also awarded and achievements recognized as a postgraduate teacher, SPPU, reviewer for scientific and scholarly journal, Computer Biology and Medicine, Journal of Molecular Graphics and Modeling, International Journal of Molecular Science, and many more. Sir has published many more international or national publications, has got a citation 824 and H index 12. We have such eminent, renowned and dynamic personality with us. Now I request Dr. Rajendra Bhadane sir, please guide the delegates on the topic of biocatalysis and ADP-ribosyl-transferase and introduction to single particle protein crystallization. I am sure we will be enriched by this session. Once again, I welcome all of you for this session. Sir, please.
Thanks, Raman, for a nice introduction. And thank you, M.S. Gosavi College of Pharmacy, for inviting me for this e-FDP session. So now I'm presenting, okay, just to heads up, can you see my slide as a full screen?
Yes, sir.
Okay. So today's topic is biocatalysis of ADP-ribosyltransferases, and I will also talk about introduction to single particle protein crystallization. So first of all, ADP-ribosyltransferases. So what is ADP-ribosylation? So ADP-ribosylation is one of the post-translational modification where one or more ADP-ribose molecule get transferred to another protein. So before I go deeper into this post-translational modification, so first of all, I like to tell you about transcription and translation are the processes where first in transcription, mRNA molecule is synthesized, and this mRNA molecules then come back from nucleus to cytoplasm. And once it reaches cytoplasm, it starts to synthesize proteins using amino acids. Transfer RNA transfers, and then ribosyl RNA arranges these monomers of amino acids, and then the protein chain is synthesized. That's called a transcription process. And once the transcription is over, then comes the translational. So transcription is like a transfer of information, and then translation. So then translation, the protein is synthesized, and once the protein is synthesized, it's like getting showered, and then you need to modify this protein. So modify means protein then arranges itself into three-dimensional arrangement, and this three-dimensional arrangement depends on the types of amino acid present in the protein. For example, if there are tryptophan or other hydrophobic amino acids, then normally these amino acids, if they are arranged in the chain, then it normally forms a beta strands. So these structures, it's called beta sheets or beta strands. And if there is a amino acid like leucine or alanine or methanoine, then it forms the helix structure like this. So this is a tertiary structure, and this secondary and tertiary structure then arranges itself to a quaternary structure, so or a three-dimensional structure of protein. So this is how typically this ADP-ribosyl transferases enzymes looks. So they have this typical arrangement of 6 beta sheets. So S1, S2, S3, S4, and S5, and S6. And then intermediately there is a alpha helix. And this arrangement makes a cleft into the enzyme, and then in that cleft NAD molecule binds. So NAD means nicotine amine adenine dinucleotide. So this is the structure of nicotinamide or nicotinamide adenine dinucleotide phosphate. So this is not phosphate but NAD. So this we call NAD plus because there is a charge on this pyridine nitrogen. So that's why it's like NAD plus. And what is ADP-ribose is a part of this NAD. So because it is a diphosphate, because there are 2 molecules. First is nicotinamide part of this nicotinamide part bound to ribose, and then adenine part. So this is adenine. So purine, this purine ring attached to ribose and 2-phosphate. It's called adenosine diphosphate. And when it's normally when it breaks here, the bond is broken here, and it forms ADP-ribose. So this is ADP-ribose part, and this is nicotinamide part. So what is ADP-ribosyltransferase? So these are the enzymes who does two steps. It works in 2 steps. First step is NAD hydrolysis. So they hydrolyze the NAD, and that means the breaking of the bond between ADP-ribose and nicotinamide. And the second step is transfer of ADP-ribose molecule to other protein. So that's the reason they call it transferases. So the molecule get transferred to other protein. So these other proteins can be any substrate or any other GI or G alpha or G protein coupled receptor inhibitory subunits, any of that subunit. So what is the key features of these enzymes? So these enzymes are having also called as ARDS, and these ARDS having a conserved residue RSE. R stands for arginine, S for serine, E for glutamic acid. Or it can have HYE motif, means histidine, tyrosine, and glutamic acid. So it can be different. So these are the typical locations of these RAC or HYE residues on the strand. So they normally present here or here. And based upon the arrangement, there are 2 types of ADP-ribosyltransferases. So they are classified into two different subgroups. So first we will see the poly-ADP-ribosyltransferases or PAR-lating enzymes or PARPs. So these are called as PARP family enzymes. So poly-ADP-ribosyltransferases. So what it does, so poly-ADP-ribosyltransferases are mostly present in the nucleus of the cell, and they are and there is a subfamily which contains different parts. So PARP1, PARP2, pancreases, TANX1 and TANX2. And these enzymes have a conserved HYE type motif. So these are HYE type ADP-ribosyltransferases. What they do? So these enzymes are actually modifies the DNA. So when there is a single strand break happens, when the DNA strand gets broken due to any of the event, then these enzymes goes there, recognize the site, and then adds the ADP-ribose molecules. Because and then this ADP-ribose addition, so it happens by transferring of nicotinamide adenine dinucleotide or NAD, conversion of NAD to ADPR, and then this ADPR then transferred to the target, means the DNA strand. And this happens repeatedly. So there are quite a lot of ADPRs then bind to this DNA, and this leads to initiation of the process, which leads to like an abnormality, and this abnormality is then recognized, and then downstream process of DNA repair starts. So this enzyme is very crucial for DNA repair mechanism. So once the ADPR binds there, then DNA repair starts, which then repairs the broken strand of the DNA. So, and this enzyme is, as it's very crucial, so it's very important in cancer cells. So because cancer cells synthesize DNA quite often, and during that process there are quite a lot of points where the DNA strand is not formed properly, and there are quite a lot of breakage. So then PARP activates those broken points and then got repaired by repair enzymes or downstream processes like homologous repair mechanism or etc. This is about the poly-ADP-ribosyltransferases, because there are multiple ADP-ribose transferred to the substrate. That's why it's called poly-ADP-ribose. But there is also a big group of enzymes, also called as a Mono-ADP-ribosyltransferases or MAR-lating enzymes or MAR. These are PARP3, 4, 5, 4, 6, 7, then PARP 8, 10, PARP 11, 12, 13, 15, and 16. These are MAR-lating enzymes, and they have HY motif, and HY and E can be it can be glutamic acid or isoleucine or leucine, or it can be tyrosine. Subcellularly, MAR-lating enzymes are found in nucleus and also in the cytosol. So they are not fixed in one location, just like PARPs, which are mostly located in the nucleus. MAR-lating enzymes or Mono-ADP-ribosyltransferases located in cytosols. The big difference is that it only transfers a single ADP-ribose unit to the substrate. This is the difference between Mono-ADP-ribosyltransferase and Poly-ADP-ribosyltransferases. And these are the host enzymes. These enzymes are present in eukaryotes or in humans. Now I will talk about the ART or ADP-ribosyltransferases in bacteria. So bacterial ARTs can be divided into two main classes based on their conserved features, and these conserved features are three significant residues in ART fold, that is NAD binding fold. And these are, first is RAC class enzymes. So these comes under the class of chlorotoxin group. So chlorotoxin group of enzymes falls under RAC class, and HYE class related bacterial toxins are called are also called the diphtheria toxin group or DT group. Chlorotoxin group is further subdivided into chlorotoxin like then C2 like and C3 like ART, because there is some modification in these key residues. So that's why there are again subtypes. And there are also some toxins known now that do not belongs to either of these RAC or HYE class. But overall, most bacterial toxins are falls under this category. And Our the toxin which we are interested are cholera toxin, and we are also working on pertussis toxin, which falls under these classes. So mechanism ART. So how ART works? So when ADP-ribosyltransferases, how the transfer of this nicotinamide adenine dinucleotide takes place, or how these ADPR get transferred. So this is the typical structure of nicotinamide adenine dinucleotide, NAD, binds to the enzyme. Normally there is quite a lot of hydrophobic interactions here, and then there are some interactions arginine molecules, and then glutamic acid helps the ribose part here like this. So due to this, there is some strain on this bond. So there is a strain here at this position, and this strain creates the lot of energy, high energy, conformation of NAD. So this high energy conformation then leads to breaking of the bond. So when glutamic acid interacts with this ribose part, and when the substrate attacks here, the as a nucleophile, it attacks, and then it breaks this bond between this pyridine ring and ribose part, and then there is a formation of oxocarbenium ion intermediate. So this oxocarbenium ion intermediate then responsible for it's very reactive, and then it attacks, it forms the bond with this incoming substrate, which is a nucleophilic substrate, and forms the bond. Normally, most of the time, this nucleophilic substrate can be a DNA, or it can be other proteins. So if there is and this other proteins then get modified. So that's why it's so this ADPR get transferred to this substrate protein. That's why these enzymes are called ADP-ribosyltransferases, or transfer of this group takes place. So what's the story with us? So we did some crystallization of pertussis toxin S1 subunit. So this is a this is a pertussis toxin which causes whooping cough in humans. What happens when the pertussis toxin S1 subunits? It actually combined with S2 subunit. So S1 and S2 subunit plus S3, S4, S5 forms the homo tetramer, and this homo tetramer with S1 subunits attaches on the human cells, not mostly the lung cells. And when it attaches there, it transfers the S1 subunit inside the cytosol. Once it goes in cytosol, S1 get activated, and this activated form of enzymes then converts a nicotinamide adenine dinucleotide from in the nucleus, in the cell, and then transfer this ADP-ribose part to G alpha I subunit of human G protein coupled receptor. Once it binds, it forms a covalent bond, and with the cysteine residue of G alpha I subunit. As G alpha I subunit get inhibited by this toxin, so there is inhibition of the signal. So G protein coupled receptor doesn't get inhibited, and then the signal is active, and that results into coughing. So that's the reason why people with the whooping cough, that's called a whooping cough, is a symptom of this toxin, because this is how it works. So it increases the cyclic AMP level in the cell, and that leads to coughing reflexes. Past in days, we crystallized this the structure of this pertussis toxin with BAAD. So we name this new compound BAD. It's a completely new molecule which formed during our research, and we also crystallize NAD molecule, and the story starts here. So what's the story? So these are the three structures crystallized by our lab. So first is 7-SKK, 7-SKY, 7-SNE. And here is the aligned structure, so all are aligned together. So first is 7-SKK is ADP-ribose and nicotinamide. So it shows the process how nicotinamide bound and we were very lucky to get crystal structure of this. So we captured this moment, how it breaks the NAD to ADPR and nicotinamide. Then we also successfully crystallized the beta NAD with iodine molecule as inhibitor. So when we added iodine, it inhibited this process of ADP-ribosyltransferases, and we were able to get the crystals with beta NAD. And while doing this crystallization, we were looking for inhibition of this pertussis toxin by small molecules. So we wanted to have small molecules inhibiting this toxin, so then we will not get the coughing reflexes. So we wanted to develop a small molecule drug as a drug molecules against this toxin. While doing so, we accidentally got a new structure, new compound formation, and we call it as a BAAD because it has a benzamide so nucleus, and that's why we call it the benzamide adenine dinucleotide. So what happened? So we got the crystals of this enzyme with this completely new molecule, and we were interested to see like why and how this molecule is formed. Is it like it? Because most of the time when we do the crystallization is a very long process, so it takes many days, sometimes months to get crystals. And during that process, we thought that maybe something happened with NAD, and when we added 3-amino-benzamide into this crystallization reaction, we might got some artifacts, and then this is completely new molecule we form. So we were interested to see whether this is a natural process or whether it can possible that this enzyme can produce these type of molecules naturally. So then we wanted to develop an in vitro method to see whether this type of new molecule production can happen in real life, not just in crystallization. And we also wanted to rule out the possibility of crystallization artifact. So what we did, so first we developed the HPLC-based method. First we analyze, so we developed the method with the nicotinamide NAD plus. Then we also analyze 3-amino-benzamide and how its retention time with that particular mobile phase works. Then we also wanted to see how nicotinamide elutes in the HPLC and how ADP-ribose elutes. So we use the standards of all these four, and we see that the retention time is quite different for all these components. So then we went to proceed with the enzymatic reaction. But before that, we also put some controls just to see that we are not getting anything in the non-enzymatic reaction. So first we injected the buffer and we see there is nothing. Then we injected nicotinamide. So first we incubated just a blank sample. Then we had NAD and NAD molecule in the water, and because nicotinamide is not stable molecule, so it in water it get hydrolyzed naturally. So we saw that after 74-hour reaction in vitro reaction, we saw some ADPR formation. And when we have a nicotinamide plus 3-AV molecule, we see the similar reaction, plus we have a peak for 3-amino-benzamide. Okay, so but apart from these 3 peaks, we didn't see anything else. Then we introduced another reaction where we have all these 3, all this with the enzyme. So first we had enzyme, so pertussis toxin as an enzyme, and nicotinamide. Because pertussis toxin is ART, so it first hydrolyzed the nicotinamide adenine dinucleotide to ADPR and nicotinamide. So and we see that the enzyme is active and it's working. So nicotinamide peak disappeared after 74-hour reaction. We saw nothing here, and we see the peak for nicotinamide and ADPR formation. So completely new peaks forms here after enzymatic degradation of NAD. When we incubated NAD with 3-aminobenzamide and pertussis toxin, we saw these peaks along with 3-aminobenzamide, and there is a small peak observed at retention time, something around 8. So we were interested to see like what is that. So if you see this zoom out readout here, you can see there is clearly a new peak formation which is absent in control peaks. It is nowhere in the control samples. We use a PDA detector, so we scan every wavelength there. So, and we also did a peak purity scan, and we analyze the spectra of these peaks. And here we see that nicotinamide adenine dinucleotide, or NAD+, it has a typical UV spectrum like this, where the maxima is 260. 3-aminobenzamide, on the other hand, does not show any clear-cut peak. It shows at something around 220-30. It shows some one maxima, and then a shoulder here at 245, and then a clear-cut peak at 310 or 20 somewhere. Nicotinamide shows a peak at 280. And then we wanted to scan the peak for this little pillar here, which we observed in this sample. And then we see that it has a spectral pattern just like nicotinamide, and also has a pattern like 3-aminobenzamide. So we were interested and curious about what is this molecule. So we went on doing LCMS analysis. And when we do the LCMS, when we did the LCMS scan, we saw this BAD, molecular ion peak formation, and there is also some adducts with ammonia and electron adduct or hydrogen adducts we got, and the fragmentation pattern then reveals that this is indeed a BAD molecule which we observe in crystallization. So this prove that this enzyme not only forms this new completely new molecule in not only in the crystallization studies, but also in the in vitro setup we perform. And that was quite exciting because this is very rare that this enzymes other than normal ADP-ribosyltransferases process that can also synthesize completely new molecule. And hence we wanted to have, is it possible to get new molecules? So then we use pharmacophore mapping studies. So first we use 3-aminobenzamide as a starting point, because that's a molecule which leads to the formation of this compound. So we analyze the pharmacophore of this 3-aminobenzamide, and it has some features which we kept. So donor features on nitrogen, acceptor features on the carbonyl oxygen, and then ring character. And using these features, we screen and we screen the database of molecules from zinc database. So there are like millions of compounds we screened, and then we selected some of those compounds, filtered through this pharmacophore mapping tool, and use quantum mechanical simulation. For quantum mechanical simulation, because it's time consuming, so we took only 600 molecules out of those, like a high scoring molecules based upon pharmacophore hypothesis. So base scoring molecules were taken, 600 molecules, and then we went on doing quantum mechanical simulations. So in quantum mechanical simulation, what happens is like it's different than molecular mechanics or MM simulation. Quantum mechanics simulation deals with the orbital theory, and then it calculates the electron movement and around the nucleus. And after these QM simulations, we observe these HOMO-LUMO energies, so highest occupied molecular orbitals and lowest unoccupied molecular orbitals calculations. So these green and red bulbs are actually bulges. Here you can see are actually HOMO-LUMO energies. So this is the HOMO energy of this molecule, and this LUMO energy, or lowest unoccupied molecular orbital. The question arises why we are doing this, because we observe that to get a successful to to get this new molecule formation or successful formation of new molecule, we need a features where our compound should be nucleophilic, and that is an important feature is required for getting this formation of new molecule. So how we can judge whether this molecule is nucleophilic or non-nucleophilic? We can see obviously by the structure, but we need more quantitative tools. So we use this quantum mechanical simulation. So this QM simulation led us to identify whether this molecule is quite nucleophilic or not. So this bulges actually tells that there is a high electrons there, higher occupied molecular orbitals there. That means it can act as a nucleophile, and there is unoccupied orbitals are not there. So that feature, then we screen in every molecule which we had. There are quite a lot of molecules. So these are some few examples I am showing here. And then we purchase these compounds from commercial sources. And after purchasing these compounds, we went on doing the same experiment. So what we did with the 3-aminobenzamide and NAD, we did the similar reaction with all these compounds. But first of all, we need to develop the method for each and every compound. So all these compounds underwent method development. So I did HPLC method development for each compound because it's very important that this compound does not interfere in the retention time of nicotinamide or ADPR or NAD. Okay, so after making sure only those molecules which have a distinctly different retention time, those are screened here. There are 14 compounds we screened. Out of 14 compounds, there are two interesting cases which I will discuss here. So first, we injected this molecule. This is the Cas number of this molecule, and we see this is the retention time, something around 8 to 9 for this molecule. And when we incubated this molecule with just nicotinamide, we see that there is a peak for nicotinamide, adenine dinucleotide, and then ADPR formation, and then small ADPR formation, and then a peak for this molecule. And when we incubated with the enzyme, so we have a control sample here, so without enzyme and with enzyme. And in control sample, we didn't see any peak formation, meaning that this reaction does not take place just with NAD and this molecule, but it needs the enzyme. So in presence of enzyme, there is a conversion of NAD to ADPR and nicotinamide. Plus, there is formation of this small molecule compound. This is zoom out readout for this molecule, and then UV spectra of this molecule, which distinctly shows the features it shows the feature of just like 3-aminobenzamide. Similarly, we also identified this another compound which shows the same pattern. Here the retention time something around 14, and then there is nothing here with the control sample, but with the enzyme there is a formation of this molecule. Peak purity scan help us to identify and get the preliminary assertion about whether this type of molecule can be formed or not. So, and we confirm by peak purity scans, and then we went on doing LC-MS studies, and we confirmed the formation of these two new molecules, and we named those molecules accordingly. So now, okay. Now, question is why we are doing so? Why we are synthesizing these small molecules? Now, if you see the NAD field or nicotinamide adenine dinucleotide is very key molecule. During crystallization, the proteins which are active in converting nicotinamide to nicotinamide adenine nicotinamide and ADPR, they are active proteins. And most of the times, crystallization is difficult with the active form of enzyme. So we need to have an inhibitor, or we need to have a certain compound which binds to the protein and stabilize the protein. But protein should not do anything, because if the protein is functioning, then it's very difficult to crystallize the functioning protein. So either you need some inhibitor to get crystal structure, and you should be very lucky to get crystal structure in case where the protein is active. And that's the reason we want to have molecules which can be act as a biochemical tool. So these molecules which we are synthesizing, the purpose of this molecule is to get to act as a biochemical tool for crystallization or those similar things. So first we started to analyze these molecules. So first molecule, which is isoquinoline amide adenine dinucleotide, because it has isoquinoline ring structure, and the second molecule is having isoindole ring. And to study these molecules, we took the reference NADH. NADH is another form of NAD where it has it shows a very strong fluorescence, NADH. And we took that as a molecule as a reference, and we developed the method for fluorescence analysis. And here we saw that this parent compound, which we use in this reaction, has very high fluorescent molecule. And because this new molecule, isoquinoline amide adenine dinucleotide, is a precursor from has a precursor of this compound. So that's the reason why it shows a fluorescence spectra. And this is a fluorescence spectra of NADH and compared with isoquinoline amide adenine dinucleotide. And it is very distinct as compared to NADH fluorescence spectra, meaning that it has some features similar to NADH, but it's not related to NADH. Means that the isoquinoline ring is responsible for fluorescence and not the NADH part of this compound, which also confirms that this compound is successfully formed. Then, while working on this manuscript for submission, we got some questions from reviewers about the non-hydrolyzable nature of these molecules, because we claim that these molecules are non-hydrolyzable, and that's an absolute requirement for crystallization study, because the molecules should be stable during crystallization. So we wanted to demonstrate that these molecules are indeed non-hydrolyzable. So what we did, so first we analyzed nicotinamide adenine dinucleotide, and in water it shows some hydrolysis product, but it's not enough to demonstrate the hydrolysis. So we went on doing hydrolysis with NaOH and HCl. As compared to hydrochloric acid, alkali hydrolyze this NAD quite more extent. So we choose that reaction, and here you see that NAD then hydrolyzed to ADPR to more extent than just a molecule sample. So there is a hydrolysis product. So if we do the similar reaction for these new compounds, so they should also show the decrease in the peak. But here we see this compound BAD, then isoindolene amide adenine dinucleotide or isoquinoline adenine amide dinucleotide, and they shows this typical peak. And while doing the hydrolysis with NaOH, instead of decrease in the peak, we saw increase in the peak height. And this is very strange, but for us it was not, because we know that in NaOH condition, NAD get hydrolyzed. So this hydrolyzed product then forms the oxocarbonium intermediate, and that could be responsible for formation of additional BAD or LIAD molecule in the solution. And that's the reason why instead of decreasing the peak, we got increase in the peak. This also indeed confirmed that these molecules are non-hydrolyzable, meaning they doesn't get hydrolyzed in NaOH or very harsh alkaline condition as well, which confirms that non-hydrolyzable nature of this molecule. Then we wanted to study these molecules to have some biochemical function, whether this molecule have any biochemical function or not. So we took PARP10. PARP10 is a very important enzyme in cancer. So we use the catalytic domain of PARP10. So this is PARP10, and we develop a method of NAD hydrolysis method. So we express and synthesize, express this protein, PARP10 protein, catalytic domain of protein, and then we incubated this protein with nicotinamide adenine dinucleotide with different concentration. And you see that 5 micromolar, 10 micromolar concentration. We saw a decrease in quite decrease and 1 micromolar concentration, and you see the steady decrease in NAD concentration. So this is the peak for NAD without any PARP enzyme, and with PARP enzyme it start to decrease, meaning that PARP consumes this NAD and forms ADPR and nicotinamide. So then we incubated the PARP enzyme with these three compounds, but due to fluorescence nature of one of the compound, we couldn't took that compound because this assay needs to be done at 450 nanometer, and the other compound, the isoquinoline amide adenine dinucleotide compound, which we have obtained, has a fluorescence characters at the same wavelength. We couldn't took that. We could not able to take that compound in this assay. So we took only two. So BAAD, the molecule which initially formed, and then the new molecule which we have obtained after optimization or computational screening. And while doing this assay in 96 well format and 384 well format assay plate, we saw that this is the typical NADP, and then this NAD get consumed by PARP10 enzyme here at 5 micromolar concentration. And when we incubated this enzyme with BAD or this other small molecule LIAD, we saw that the consumption stops. So there is increase in the peak. That means NAD consumption is inhibited by incubation with this isoquinoline amide adenine dinucleotide, meaning that this molecule is inhibiting the PARP10. So we got a very good lead of this molecule that we are not just synthesized non-hydrolyzable, completely new non-hydrolyzable NAD analogs, but also we developed a molecule which actually inhibits PARP10, which is important in cancer. And this paper is recently accepted in Journal of Biological Chemistry, a very renowned journal in biochemistry field recently. So this is the story about this, all about this. If you have any question about this part, let me know, and then I can move to the next part. Is there any question?
So please continue.
Okay. So if there is no question, then I can move to the next part. So basics of single particle protein crystallization, because it's very routine for our lab to do crystallization studies, and we are involved with different proteins, and we try to get crystal structure of different proteins. But to do the crystallization work, it's very okay. So this is my lab here in BioCity. So, and we have this. We need a very good infrastructure for studying different aspects. So this building does not only holds crystallization studies, but also does quite a lot of other things. So there is a functional genomics unit here. Then we also have a cell imaging and cytometry, proteomics, bioinformatics, protein structure and chemistry, which is part of my lab, and genome editing, CRISPR-Cas and those. And then metabolomics studies we do. And then we also have a zebrafish facility. We have single cell omics technologies and screening technologies. So this is all part of this big BioCity companion. And here we do protein expression. So for crystallization, you need a protein, and how to get the protein. So first of all, you need to identify the gene of interest or a protein of interest. So that protein of interest means that it comes with the genomic sequence. So identifying the sequence and then designing the strategy to get the clone. Once you get the clone, then you need to insert the clone to suitable vector. So vector or we can call it the plasmid. So you need to select the proper plasmid and the gene of interest. Once you identify, you can clone it. So we have a facility for not just for synthesizing DNA. So we have a PCR, different qPCRs instruments. We can synthesize the DNA, and also we have quite a lot of vectors in our lab. So we do the cloning, and once the clone is successfully inserted into plasmid, then we have certain cell lines. So these are bacterial cell lines. So this is BL21 cell, BE3 class cell. This normally has T7 expression vector. So once we transform the plasmid containing gene of interest into this bacteria, then bacteria starts to produce this protein. So how does it work? So first of all, you need to have expression vector with the gene of interest, and then you also need some antibiotic resistance gene. So antibiotic resistant gene is for so for example, if we have kanamycin resistant gene, so then we incubate this bacteria in a media, LB media or auto induction media, and when we incubate the bacteria into the media, we also add antibiotic. So adding antibiotic then ensures that all other bacteria present in the culture get dead, and only the bacteria which has this plasmid of interest survives, because it has a gene for resistance against that antibiotic, for example, kanamycin. So this is how we need to ensure that only the bacteria which is producing the protein of interest is surviving, and not the other all other bacteria. So this is a typical way of recombinant protein expression. Because for protein crystallization, we need a protein in milligram scale. So we need to incubate in a quite lot of big lots. So we need to have this type of incubators, big incubators, to grow the culture. So normally we incubate in 4-liter flask cultures, and after incubation, then we need to do the lysis. So lysis means once you grow the bacteria which produce the protein of interest, then you then broke the bacterial cell membrane and release the protein of interest. So that's done by lysis of that bacteria. So that bacterial lysis is normally done using sonicator method or emulsification method. So we have both instruments, emulsifier, and also we have probe sonicators, different types of probe sonicators to sonicate and lyse the bacteria in appropriate lysis buffer. Once the lysis happen, then the protein is then released into the solution along with the genomic impurities and the cell membrane and everything, and also other proteins. Then we need to remove all these derbies out, because because we expect that protein is in a soluble form in that particular buffer. So then we use this high-speed centrifuge. For example, this is a Beckman counter centrifuge. We first centrifuge the bacterial lysate, and after centrifugation, we get the filtrate, which is then used for protein purification. During this process, I forget to mention, because here when we insert this clone, we also insert some tags. For example, his tag or GST tag. So these tags are actually are additional sequence of amino acid. For example, currently his tag purification is very widely used method. So what happens, like 6 histidines are added along with the gene protein of interest, and this gene can be added at N terminal or C terminal, depends on functionality and how the tag can affect the protein function. So what happens? So when we have protein of interest plus his tag, 6 histidine residues or his tag. So this his tag, then we use nickel NTA. So nickel NTA is a one of the purification, IMAC purification, that's called IMAC purification. So it's a metal ion affinity purification, meaning that when we have beads containing a nickel ion with for example histidine or histidine residue, then once we allow this lysate to pass, the clarified lysate which we centrifuge, when we allow to pass through the column, then the proteins start to interact with the nickel ion because nickel has affinity towards histidine molecules. So 6 histidine tags ensure that all the proteins in the solution get binds to that. And those proteins which don't have his tag, because bacteria when expresses the protein, it also expresses its own proteins also. So the solution not only has your protein of interest, but also all other proteins. So we have our protein of interest plus some residual proteins. So we need to get rid of all those proteins. So for that, first we do the his tag purification. And then in our lab, we use this type of purification method. This is called ACTA purifier. So this is FPLC or FPLC instrument, which is typically used for protein purification. So it can host number of columns. So 6 columns can be added. So first we add his tag purification, and once we got his tag purification, then we need to do sec purification or size exclusion chromatography. So size exclusion chromatography is based upon the size of the proteins, because every protein has a different size. So when we allow this protein his tag purified proteins flow through this sec column, it's a second stage purification just to get more polished protein and remove any other impurities which might come after nickel NT purification. So then sec purification does this, and also this crystallization works needs very, very pure protein. So protein needs to be pure more than 95%. So that's why we sometimes need a two-step purification protocol. That's a requirement. So we use sec, and this sec purification then ensures that we get proteins by the size. And then if we know that sequence of the protein, based on that we can easily calculate and where the peak is appeared. So we collect the fractions and then analyze the fraction. Typical analysis is done with the SDS page. So this is electrophoresis unit. So we use this sodium diadesyl sulfate polyacrylamide gel electrophoresis or SDS page. So here is a SDS page gel. So for the characterization, we add the samples containing the whole lysate, and then sample with the purification method. And so, for example, this is a nickel NT purification where there are some other impurities also present, and then there's a sec purification, and we also have a ladder containing the known size bands. So we know what is the size of the protein based on the reference. So most of the time we do a normal SDS page, but sometimes when we express a completely new protein, so we need to identify whether this expressed protein is the right protein or not. So we need to do Western blot analysis. So Western blot is a extension. So first we do SDS page, and before we develop this gel using Kumasi staining, so we don't stain it, we use the gel and then transfer the gel on the paper. So this is the blotting paper. So this is how Western blot takes place. So we take the gel and then nitrocellulose membrane, and then overnight incubation at 12 volt. Normally we have a protocol that then does the weight transfer in the Pharmacophore, leads to transfer of protein from gel to the nitrocellulose paper. And once the gel get transferred, then we add a primary antibody to react with the to form the complex with the protein, because this primary antibody is very specific. For example, it's specific to his tag, so it only binds to the his tag containing proteins, not to any other protein, and then we add a secondary antibody which gives the color reaction using a chemiluminescence, and that confirms that this is the protein of interest and it is successfully expressed or not. So this is how we need to do the characterization. Once we confirm that we express the protein, right protein, and once we got the confirmation and once we got a pure protein by both nickel NT and after sec purification, we need to do different characterization. So first, this is one of the instruments which normally we use for differential scanning, fluorimetry scan. So this is called nano DSF. Nano DSF is useful for protein folding and protein folding analysis, and also for calculating the stability of the protein. So what happens? So in nano DSF, the protein is placed into a small capillaries, like 10 microliter balloon, and these capillaries are scanned at different temperatures. So it starts from 20, for example, then 20, 21, up to 95 degrees Celsius. And then we get the graph from the instrument which tells what is the melting temperature of the protein. Based on the melting temperature, first we get the idea about whether the protein is properly folded or not, because if the protein is not properly folded, then it shows a flat DSF curve. But if protein is properly folded, means then it shows the change in the orientation of tryptophan residues and tyrosine residues, because tryptophan and tyrosine residues absorbs at 280 nanometer. So this instrument is working on intrinsic fluorescence characters of these tryptophan and tyrosine residues, and then if these residues are inside in the protein, and when the protein start to heat up, it opens the structure of the protein, which leads to the change in the fluorescence. So either there is increase or decrease in the fluorescence. That leads to the inflection point where we can calculate what is the melting temperature of the protein and how protein get unfolded or refolded. Then this is a SPR or SPR instrument, which is used for protein-protein interaction analysis and also for protein small molecule interaction. So because most of the time the crystallization takes place with it is needed to find out a new molecule or inhibitor compound. So we need to do some SPR analysis, two proteins together, or sometimes protein with a small molecule. The SPR helps us to identify the affinity of the protein with that molecule. Then there is this mass photometer instrument, which is used for determination of accurate mass of the protein. So any protein which has a molecular weight more than 30 kilodalton can be easily detected in this instrument, and it has very high sensitivity, meaning a sample containing for around 30 to 100 nanomolar sample can be easily detected, and molecular weight can be easily calculated using this instrument. So we normally use this instrument to get the accurate size of the protein. Then we also use some of the assay technologies, like this is a typical alpha screen assay for finding the protein-protein interactions and also protein small molecule interaction. So if there is a protein is bound to the donor and acceptor beads, and if there is an interaction between two proteins happens, then there is an enhanced signal. But if interaction inhibited, then the signal drops. That's the indication that this protein get bound to other protein or not, or whether there is inhibitor molecule acting on the protein or not. So that can be confirmed by this alpha screen assay. Then there is also this another instrument which is called a SEC-MALS, so size exclusion chromatography coupled with MALS. So MALS is multi-angle light scattering detector. So this is typical wired instrument, MALS detector. So this detector base is actually works along with UV detector. It has a light scattering detector and refractive index detector. So what happens when the protein goes through this both instrument, then it first gives the UV signal for the UV signal for protein at 280, and then we get this signal for light scattering. And this light scattering can be forward scattering, back scatter, and the front scatter, and then a refractive index, which is another universal detector. Based on the light scattering, we get the very accurate size of the protein using this instrument. So that's very important. And also, if there is a protein-protein interaction happens, then molecular weight change takes place, and that can be easily assessed by this SEC-MALS instrument. This instrument is also very useful for to study the batch-to-batch variation, because sometimes for crystallization you need bigger batches, because you need to attain a certain concentration. So you need to purify proteins number of times, and to get the quite batch-to-batch variation known, then we need to use this type of instrument. Then this is another instrument which we normally use for during crystallization. Like this is MST instrument. So it's microscale thermofluorescence technology, where the IR light is passed on the protein plus a substrate molecule. It can be another protein or it can be small molecule, and then based upon the thermal scan, it detects whether the protein is bound to the small molecule or not. And based on that, it gives very nice IC50 values for these compounds. If we can't do MST, then we also have this ITC, isothermal titration calorimetry instrument, for detecting or for the binding analysis. Why these things necessary? Because protein crystallization, as I told you, it needs a protein with a small molecule, because most of the time protein gets stabilized with a small molecule, and most of the time our aim is to find how protein interacts with these small molecules, and based on that, then we can screen the compounds. So that's the reason why we use all these instruments. Then actual crystallization, how it happens. So normally when we start the crystallization thing, so first of all we need to screen the different conditions, different buffer conditions, because protein cannot just crystallize in a random buffer. Or it actually does it randomly, but it's very difficult to predict in which buffer this protein will crystallize. So you need to do the screening. So screening means it's called buffer screening. So this is a typical format of 96-well format plate. So this one block looks like this, and this block has one reservoir where we have a screen solution. So we can add, for example, 50 microliter screen solution, and there are 3 points, 1, 2, and 3. So here you can have different ratio of screen solution to protein. So normally we have like 50. So here typically we add like 50 nanoliter protein and 150 nanoliter the screen solution. Then here we can add, for example, 100 nanoliter protein and 100 nanoliter screen solution. And here another drop containing protein, maybe 150 nanoliter protein and 50 nanoliter screen solution. So this is how we use a single buffer and in 3 different ratios. The same is repeated again on all the 96-well format. So this way we get 384 different conditions for that particular buffer conditions. So one buffer and 3 different conditions. So this way we need to screen different screens. So there are different screens available in market, like Molecular Dimension, or Hampton Research or Jena Bioscience. If you go through this website, they have different screens. So for example, JCHG Plus or PAC Premier or Morpheus 1, 2, 3, or ligand strategy screen, or MIDA screen, or index screen. So there are Helsinki Cryo or other screens. So there are different screens are available. One screen condition can lead to 96 different buffer conditions, and those can be screened. So once we apply, so because this is typically nanoliter solutions, which is impossible to pipette out using your own hands. So you need to have some robotics. So we have these pipetters, robotic pipetters, which pipette out the solution. So we just need to put the reservoir solution in the wells, or that can be done also by the robotics. So we have those facility here in our labs. Once we plate the protein, we need to seal this this plate using this sealant, and that sealant needs to be transparent for scanning over the time. So this way the protein and the plate is then sealed, and after sealing this plate like this, then we put this plate into this rock imager machine. So this rock imager is another robotic instrument which periodically takes the plate out and then scans the plate for not just for visible light but also UV light, and then it screens the and it try to see whether there is a crystal formation or not. Once if you are lucky enough, if you get the crystals, so the crystals can be like this or like this. So this is the drop protein solution. For example, this is 200 nanoliter drop, and in that you might see this type of small crystals. There may be some needles. Once you confirm that you got the crystal, so you start to you can use the this pH scanner. So this is another instrument available in our lab, which is used to differentiate whether the crystal formation happened due to protein or it is a salt crystal, because most of the time these buffers contain salts, so they can also get crystallized. So what this instrument does, this is an X-ray scanner. So it emits X-rays and scans the plate. So the big benefit of this instrument is that you can load the entire plate. This plate can go from here inside, and then the protein is screened directly without taking out from the drop. So this is in situ detection, and this then detects this type of crystal image. Then the phase diagram is created, which then help us to identify whether this protein is a salt crystal or whether this is a protein crystal. Once we confirm the protein crystal, then we need to use with the in-house X-ray crystallography instrument, which collects the data for the protein, or we send these crystals for high resolution crystallization experiments, which is done at synchrotron facility. So for that purpose, we need to first fish the crystal. So that's called a fishing exercise. So this is typical fishing hook. You can see here. So we need to fish this crystal, which is using a microscope. So under microscope, you need to carefully fish the crystal and immediately transfer this crystal into this type of puck. This is called unipuck. So this unipuck is magnetic in nature. So, and this loop is also magnetic. So once you hook the crystal into this fishing hook, then you just drop it here, and it get locked. This done in liquid nitrogen. So this is liquid nitrogen, and this puck is placed into liquid nitrogen, immersed into that, and you just immediately freeze the crystal in that particular state. And once you freeze that, then you can have a number of unipucks that goes into this DVAR flask, and then you ship this DVAR after you ship this DVAR flask to the synchrotron facility. So I can show one video how it is done. So for example, these are the unipucks. So these are the fishing hooks. And okay, let's see. Okay, so this is how typically it's done. And once you phase the crystal, you need to send this to synchrotron facility. So synchrotron radiation sources, there are different synchrotron radiation sources. Here is the example. So this is a ESRF ID-23-2 beamline. How much time I have? How much time do I have?
So 15 to 20 minutes.
Okay. So this is a typical synchrotron source. So this is ESRF building located in Grenoble, France. And here the X-rays are rotated. It is not X-ray, but electrons are rotated in a big circle. So it's a large. It's called a large hydrogen collider, which generates the electrons. And when electrons move, and here electrons move very fast in this synchrotron. And once it moves like this, and when the electrons are released in a tangential direction, then it leads to formation of X-rays. So this is the technology called synchrotron. So, this is a ID-23 beamline, fully robotic instrument, which can then it can then take the perk and then take individual protein fished into that individual crystal fished into that hook, and then analyze that because this is because the radiations is very intense, so it can scan the one crystal within 5 minutes. So you can get more than 3,500 images of your protein. And those images looks like this. So this is typically a phase diagram created after scanning the protein. So this is a very nice looking phase diagram for the protein. And from these phases, you need to calculate electron density. And after electron density calculation, then you use the MD simulation and other like a COOT programs, different software tools to build the atomic model. And once you build the atomic model, fit the molecular structure, this amino acid structure into that density, electron density, you get the final structure, which is a 3-dimensional structure of the protein in your screen. So this is how the journey of your protein, which is in the clone and in the lab, you express in the lab in a solution form, and then it transforms into the 3-dimensional structure, which you can see on your computer. Okay. We also have a facility for Cryo-EM and TEM. So what we do because this is another technology, this is different than single particle protein crystallization, and this is currently Cryo-EM is got popularized because of its capacity to crystallize not crystallize, but capacity to determine the structure of large molecular structures, and also it can be used to, for example, the proteins which are very big in size. For example, typically this Cryo-EM technology is very suitable for proteins having molecular weight more than 100 kilodalton. So what we have, this blow discharge instrument, and then we have this cryo setup, and we have a TEM. So first we do charging the grade. So normally we do negative imaging of the protein using a TEM facility in our own building, and once we observe the particles and the concentration adjustment, we then do the cryo screening. So in this case, this type of setup is put into this place, and then because this needs to be done in the liquid nitrogen, so this type of holder is placed in the cryo structure. And then this is a small grid here, located here. And on this grid, the protein is then frozen directly. So this is a different technology. So the frozen protein is then put into this type of dewar flask, and then shipped to this Cryo-EM facility. So we have a facility in Helsinki for Cryo-EM microscopy. and there is also Cryo-EM facility in Grenoble, France. So we can freely send the sample there and analyze the protein using Cryo-EM technology. So yeah, so this is all about the ADP-ribosyltransferases topic I speak, and also about how protein crystallization happens. I like to thank my lab members, my supervisor, Arto, Anbu, Mona, Madhukar, Sujit, and also like to thank my previous supervisor, Auti Salohen, my PhD supervisor, D. Mishram sir. I also like to thank University of Turku, Abo Academy, Academy of Finland, Novartis Foundation, Instruct Eric, Jen Athos Eric Foundation, Serenska Cultural Fund, Dentor, Board, Penty Board Memorial Foundation, FICOR, and CSC for excellent computational resources. And thank you for MS Gosavi College of Pharmacy for inviting me and deliver this session. Thank you. Yeah. Okay. If you have any question, please ask.
Thank you so much, sir, for a wonderful session that you have explained your research. So we have one question from Rebella, who says that excellent work presented, sir, but please highlight the challenges faced during such study.
The most important challenges are related to crystallization because it's highly unpredictable in which particular buffer condition the protein will crystallize. But it's always a trial and error. So, for example, we are working with one of the protein from last 2 years, and we haven't got any crystals. So we screen all the conditions, and we haven't got the crystals. So then we need to change the strategy. For example, first we either change the buffer conditions, so we use different screens, try to say use those screens. But if it doesn't crystallize, then go for the next type of screen. Then change the ligands, concentration of ligands to protein ratio. That's the one thing you can do. Then one more is to get the purity, like to know that how much pure your compound is, protein is, and check the batch is proper or not. Then you can also use this MST or other assay methods to screen other molecules which might crystallize that protein more effectively than the molecule which doesn't. But overall, it's not that simple. So sometimes you just get the crystals just out of the blue, and then, or sometimes you never get the crystals. So yeah, yes, sorry.
Thank you, sir. So do you face any problem regarding stabilization of protein structure? Because proteins are highly unstable. So they may get changed their structure during so much long procedures that you have explained.
Yeah, we need to maintain the protein. So normally we have freezers here. So we maintain the protein in -80 all the time. So whenever we need a protein, so we aliquot the protein. So we never freeze thaw the protein. So freeze thawing is the one thing which normally leads to destroying the protein, because we have protein with like milligrams quantities like 5, 10, 20, 50 milligram protein. So then if you take the protein out of the -80 and then work on the protein, if you put back that protein, it will not having the same conformation. So then to make sure that each time you have the same type of protein, then we aliquot the protein in small batches like 10 microliter or 20 microliter. So then we never put the protein back again into -80. So once we use it and we destroy it. Yeah.
Presentation. Okay. Thank you so much, sir. You have nicely discussed your research with us and giving and sharing your insights on Pharmacophore mapping. You have also focused on LUMO and HUMO concept of developing ADP-ribosomal transferase based biocatalyst of non-hydrolyzable NAD analog. So thank you so much also explaining the single particle protein crystallization using various instrumental facilities that is available with you. till the isolation of protein, till its structural identification, 3D structural identification. And also you have explained various instruments like TEAM. Thank you so much, sir. Please accept our certificate as a token of love.
Thank you.
Take this opportunity to thank you for giving or sparing your valuable time with us from your busy schedule. On behalf of Gokhale Education Society, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, all the faculty, all the delegates, on the behalf of all the delegates, I thank you for being with us and for sharing your valuable knowledge with us. Thank you so much, sir.
Thank you. Have a good day.
Okay, have a nice day, sir.